dermatologist working presently at SVS Medical College Hospital and Sri Amrita Clinics, Mahbub Nagar, Telangana. I am really happy to be the convener for this webinar on Epidermolysis bullosa, chaired by Professor V.R. Rao sir, and we have the very ace researcher Dr. Vamsi Krishna as today's guest speaker. Welcome, Dr. Vamsi Krishna. Welcome, Dr. And welcome all the participants. So just to begin, I saw my first Epidermolysis bullosa patient right in my first year of practice in 1999. Madam, could you please post that picture of my patient, first patient? It is with consent of his parents.
Yes, sir. Just a second, sir. Showing that now.
So he is late Master Sai Ganesh Verma. Unfortunately, we lost him to the disease in 2008. And today I propose to dedicate this meeting to late Master Sai Ganesh Verma and all our EB patients of all of us. Thank you for allowing me this. So, Epidermolysis bullosa, as we all know, is a rare inherited genodermatosis characterized by blistering of skin and mucosal fragility with minimal mechanical stress and heat. So all the molecules and their mutations involved in Epidermolysis bullosa are just spaced in a narrow space of the basement membrane zone or dermoepidermal junction, which we call it, which is just across 200 micrometers in width. So all of the pathology of Epidermolysis bullosa is located here. So according to the recent consensus classification, there are 4 main types of Epidermolysis bullosa, as we all know. The simplex variety, EB simplex, junctional variety, EB junctional, the dystrophic Epidermolysis bullosa, and the Kindler Epidermolysis bullosa. The most common being the Epidermolysis bullosa simplex variety. And the rarest is the Kindler variety, of which only 250 patients are there worldwide right now. So with this background, I would like to request Professor Rao sir, please post his CV, madam. So just I would like to introduce, Dr. Professor V.R. Rao sir. He is presently the Dean of Genome Foundation, which is doing great research in this area, along with all the dermatologists of Telangana and Andhra Pradesh states. The CV slide, please. Okay. So he has started his career as a research officer at National Institute of Hematology, ICMR, Mumbai, and then he worked at the Center for Cellular and Molecular Biology, CCMB, Hyderabad. And then he was joint director and director of Anthropological Survey of India, Kolkata. He was a professor at the Department of Anthropology, Delhi. Presently, he is holding the position of Dean Research of Genome Foundation, Hyderabad, and visiting professor, Thalassemia and Sickle Cell Society, Hyderabad. He has many awards to his credit, including the award received from Dr. Abdul Kalam, late Dr. Abdul Kalam, President of India. He was twice elected President of the Indian Society of Human Genetics, and he is the member of the Human Genome Organization. Sir, please give your opening remarks.
Thanks, Dr. Sharchandra, for your kind introduction and request to chair the session. Friends, good evening to all of you. I am honored to chair this session where Dr. Vamsi Krishna, a physician cum scientist from a reputed Government of India research organization, Institute of Genomics and Integrative Biology, popularly known as IGIB, is speaking. In terms of translation of genetics in clinical practice, we are witnessing success in diagnosis and prognosis of rare Mendelian syndromic, non-syndromic diseases for better management. But therapeutic intervention requires integrated biological approaches. IGIB is created with such an aim. Genomics of skin diseases was one of on their agenda. Dr. Shiva Subbu and Dr. Vinos Keria have produced several PhD theses and publications in this respect. Translation requires multidisciplinary approaches, and application of translation genomics is ultimately in clinical practice. I am glad Dr. Vamsi, being a physician, the two dermatologists, come scientist, he can make a difference in clinical application of genomics in genodermatoses. Rightly, Dr. Vamsi chose to speak on translational genomics. It will be a great evening for all of us to listen to him and learn. Friends, now may I request Dr. Sarath Chandra to introduce Dr. Vamsi and conduct the proceedings. Thank you.
It's my pleasure again to introduce today's guest speaker, Dr. Vamsi Krishna Yanamandra. He's just 36 years and has a mountain of achievements and accolades and honors and awards to his credit. He has done his MBBS from YSR University of Health Sciences and also PhD in dermatology and venereology from All India Institute of Medical Sciences, Delhi. And he has done postdoctoral fellowships and certifications from Stanford University School of Medicine, California, USA, and University Medical Center, Groningen, Netherlands. He is a visiting researcher at the King's College London, United Kingdom, an honorary clinical fellow at Birmingham Children's Hospital, UK, Guy's and St. Thomas Hospital, London, United Kingdom. He is also a member of IADVL, European Association of Dermatologists and Venereologists, and member of the European Society for Dermatological Research. He has been mentored by greats like Professor John McGrath, Professor Celia Moss, and Professor Sethuraman, to name a few. He has about 21 publications, 11 abstracts, and 2 book chapters to his credit, and guides PhD scholars and project fellows in research at IGIB. He has several awards, and if I start counting the awards, I think we will finish our 45 minutes program. Including one Professor Premlata Research Award from IADVL, because it's pertinent to us. So with this short introduction, I request Dr. Vamsi to please start his talk.
Thank you so much, sir, for the kind introduction and the invitation. Shall I share my screen?
Yeah, I think it's right now. Yes.
My screen visible? Is my screen visible?
Yes, yes.
And I'm audible?
Yeah.
Yes, sir.
Oh, perfect. Thank you, first of all, for the invitation, sir, Sharath sir, and Dr. Professor V.R. Rao, and Genome Foundation for conducting this successful webinar series. Yes, as already introduced, my topic is on translational genomics and genodermatoses, specifically on Epidermolysis Bullosa. So I thought I will just begin with a couple of slides with EB to just get over it with the introduction and then focus on more important things. So basically, as Sharath Chandra sir has already showed that.
Sir, one thing, sir. One small thing, sir. Sir, little more. Can you sit up and be a little louder? Little. The volume is coming softer. Sorry.
Okay. Can you hear me now? And you can
yes,
hear.
Sir. Yes, sir. That should be fine, sir. Thank you so much, sir.
Perfect. Thank you. So as EB is a clinically and genetically heterogeneous group of disorders, yes, while one considers it to be a skin-only phenotype, there is increasing evidence that it is more like a systemic disease with much more systemic manifestations. As Sarah has already mentioned about the consensus classification, this has come out recently in 2020, and I think this is what we can currently follow for EB. 4 subtypes, 16 genes involved, and multiple other subtypes that are there, close to around 33 subtypes that are known. While the burden is, there is variable clinical presentation, and because of systemic manifestations as well, it is not only managed by dermatologists. It requires a team of multidisciplinary professionals, ranging from surgeons, pediatricians, respiratory physicians, orthopedicians, including psychologists. A lot of multidisciplinary management is required for these patients. There is high mortality, specifically for junctional EB, when these kids die by 6 months of age, and we still don't know why. And most of the surviving patients do have a contracted life and at an increased risk for developing squamous cell carcinoma. Unfortunately, there is no cure still. There are certain leads that have been already developed, but until then, it is mostly daily management with wound dressings, emollients, and some off-label drugs, leading to a poor quality of life. It is more with severe disease, especially girl children, and requiring daily care. Hence, especially for diseases like Epidermolysis bullosa and other Genodermatoses, making a precise genetic diagnosis is therapeutic in its own right, because it can first enable prenatal counseling and genetic counseling. Second, it can all the developing therapies that are there. They can, these patients, once they are genetically characterized, they can be precisely included in these clinical trials. And finally, they could provide new leads to develop better therapies, which are much more affordable. Traditionally, the diagnostic approach has been to having the child suspected with EB. You look at clinical features, you make a differential diagnosis, and then you take a skin biopsy, do immunofluorescence labeling for them. And in inconclusive cases, you kind of use electron microscopy now. The idea is to identify a candidate gene that you can sequence out to find the mutations precisely and then take them to genetic counseling. But recently, with the boom of next generation sequencing, this has now been replaced mostly. And very recent guidelines have been published in 2020, as well in BJD, that encompasses all the diagnostic approaches that can be laid out for Epidermolysis bullosa. While that is there, what is the global status? I think for EB specifically, there have been clinical networks that have been developed. Dedicated patient organizations, both in US, Europe, and mostly in Western European countries, they have been actively funding own research that is directed more towards patients. And there are more industry partnerships as well, where leads from academic centers are being taken up and they're being converted into therapies. Of recent is the first FDA-approved topical gene therapy for dystrophic epidermolysis bullosa. This is through a company called Crystal in collaboration with Stanford. I'm fortunate to have worked with all these clinical networks, patient organizations, and these industries, and led some of their clinical trials, both in Europe as well as in the US. Well, this is the global status. Where is India? So in India, in general, we still have a lack of general awareness of genetic diseases. We are still evolving. The major problem that we currently have is a lack of accessible and affordable system for genetic screening or diagnosis of genetic diseases. And once you identify, while academic centers, some companies are now cropping up and multiple other avenues are being available now, there is still a lack of understanding of mutational spectrum. Recently, the government of India has released a national policy for rare diseases. Unfortunately, Genodermatoses do not figure out in them. Well, with this status, with this introduction, I just want to start my story now. Why did I even do what I'm doing currently? Well, as already introduced, my journey, if I have to summarize the entire journey, is I never wanted to be a doctor. I wanted to be an engineer. So I am an engineer, wanted to be an engineer, became a doctor, wanted to do cardiology, but ended up doing dermatology, wanted up to be a practicing clinician, but ended up being a physician scientist. Where this journey is something that I have not planned for at all. And it just happened because of what I have encountered. at AIMS during my initial days. So once I got into my residency, I was sure that I did not want to do dermatology. I was not interested in dermatology because dermatology for me at that point in time was more about hair fall, acne, and all of it. It did not appeal to me. And cardiology was where I was naturally inclined towards to. So I thought I cannot prepare again. So I need to go abroad and do residency in US. For that, I needed research experience. So I joined with Professor Sethuraman for some research experience. And he normally sees all these Genodermatoses patients, being a pediatric dermatologist. During that time, most of the patients that do come, it is like the counseling was, this is a rare disease. We do not do diagnostics also. There is no genetics that are available for them. In severe cases, the patient is going to die. Please enable genetic counseling. And this constant thing that we keep on counseling, the parents on the other side, they will keep on telling that if the father is there, she would say that the mother had an illicit affair, or the mother says, my husband is an alcoholic or whatever, that is the reason why we have this. Or the in-laws will keep on telling that some customs, like you see during eclipses and all of it, are the reasons why we have this genetic diseases. Apart from all this, there was a constant helplessness that I had because I always felt that I was a good clinician, I was academically good, and being at the best college in the country, the best that I can do was there is nothing much we can do. So hence I actually wanted to make a difference. I did not know what I could do. So normally everyone's journey is research. They have a question, they know how to do it, and then they kind of do it why they are doing it. So for me, my journey started on the opposite. Why I knew why I wanted to do it. I really did not know how I'll do it or what I should be doing. And then it started with what is the current picture with EV when I started. Diagnostic techniques were limited. There are few centers who are catering to immunohistochemistry, immunofluorescence. Management is incomplete. Research at that point in time was unknown, at least to me. And in a child's aspect of EV, you actually do clinical assessment, and sometimes the diagnosis is correct, sometimes the diagnosis is inaccurate, and management is more often incomplete. At that point in time, in the literature, we have two papers. There are good reviews that are written by Dr. Rashmi and Dr. Raghvinder Rao, who is in Manipal, who currently deals with immunofluorescence for many of these patients now. Then is when I have taken a PhD journey, and my PhD began with very simple things. Being a clinician, there was not many expectations from what I would achieve. So my goal was only to do skin biopsy, compare the regular immunofluorescence technique with a immunohistochemistry technique. Basically, try to use normal microscopy, and can we actually find it, as was my entire thesis. So this was supposed to last for 3 to 5 years. I finished it in 6 months because of whatever work that I could do. And then in December 2013, there was this international congress that was hosted by my institute, and in 2013, so where I wanted to meet Professor John McGrath, Professor Celia Moss, and another professor from another professor. Unfortunately, Professor John McGrath, he being busy, he comes, he leaves. And the other professor, I meet her after lunch, during just before lunch. We just take a selfie. She congratulates me that you're working on EB, good to see, and all of it. But I couldn't talk much. I wanted to learn more from them. So then Celia Moss gives me time the next day in the lift lobby before she goes to Agra. And I was like, how do I tell them that I want to learn more? And I have just 5 minutes to actually tell what I wanted to tell, what I'm feeling. I really did not know what to do. So next day morning when I go, as usual, Professor Sethuraman comes late. And then I walk up to Celia Moss, introduce myself, and say that I explain all the work that I was doing. And it went on to 30 minutes. And then she stops me. I was like, I spoke a lot. And then 2 days later, I get a mail at Vamsi, why don't you come to UK? I would like to train you. So it was the beginning of what I think is a pleasant journey. And at the end of that entire visit, I had the honor to meet a professor in King's College London, where we normally look up to him. He's an Indian, and he said after hearing to my thesis, he said that, Vamsi, do you even know what a PhD thesis is? You Indians are only good at copying things. immunohistochemistry and immunofluorescence are already known from age old. From 1990s, we have been doing it. And you are saying that it is a PhD thesis. Do you even know what a PhD thesis entitles to? I was dejected. I was visibly emotionally overwhelmed. And then John McGrath puts his hand and says, listen, what you are doing whatever you could do to the best of your ability, but what he says also makes sense. Just think about it, but don't lose heart. Continue your work. It was 2 hours right from my travel back from London to Birmingham, from when I'm taking my return flight, was when I asked myself a question. If I have to make a difference for epidermolysis bullosa, it is not by following the conventional route. I had to do something else. So my first question was, how can I improve my diagnosis? As Indians, I feel that the greatest challenge that we had for EV, for clinical diagnosis, was the great phenotypic variability that exists across subtypes. Right? You have a mild phenotype, as in Weber cocaine, earlier known as Weber cocaine, now EV simplex localized, to a much more severe subtype, which is now earlier known as Haliku-Simmons. Now, this subtypes, because the clinical presentation is so variable, you cannot normally get an accurate clinical diagnosis. So my question was, can I apply the same technique that we as clinicians use to identify diagnosis, that is not only looking at clinical features that are important, but also clinical features that are non-existent? Thus came up this clinical diagnostic matrix that we published. It is an algorithm of using only clinical features that accurately identifies the type of EB in close to around 92% of the individuals. And most important component is, with the evolution of next generation sequencing, it was envisioned then that it would also help in AI and machine learning algorithms to come up in the future for better diagnosis. So we converted that into an electronic application, and this is freely available both on the BGD website and then on a patient organization as well. So basically what it does is, you enter the patient data, you have all the possible clinical features that are present across all the subtypes of EB. And the principle is that you would score every cell of that algorithm, of that matrix, with either 1 or 0. So basically you're converting every clinical feature into binary digits. The principle is that it's not only important to see what is observed in the patient and what you think has to be present, but also what you did not see and what should not be also. That is the key for the matrix scoring, that both of them you give a score of 1, rest everything you give a score of 0. Basically, you add up all the columns, and the column with the highest score is the diagnosis. If more than 2 are scored equally, you would label them as inconclusive cases. So there are pictographic examples of what you can select, and once you select them, the matrix calculates all of them automatically into zeros and ones, and the ones all get added up, and the column with the highest score becomes the final diagnosis. There is an algorithm in case of multiple high scores that are available, where more specific features, like for example, poikiloderma, more specific for Kindler syndrome, lack of mucosal lesions or scarring or poikiloderma would point to more simplex variety. Or if you have excessive granulation tissue, it would point more towards junctional EB. So there is an algorithm that would clear out if there are more than 2 columns that are there. Finally, you can print that report and give it back to the patient. So this is our clinical diagnostic matrix that we, I think, is pretty much useful, and because of the utility, EB Clinic, which is a clinical network of all the centers that are catering to epidermolysis bullosa, they have hosted it on their website, and this is freely available for every practicing clinician, not only dermatologists, pediatricians, everyone. They can just freely download it and use it for their regular practice. This is currently used in multiple countries, and I am always proud to show off this. Sorry about that, but for this, in 2014 was when I went to UK, and 2015 was when I won this award. And this was the first time an Indian has won this award in its 93 years. This was the original award. Because they could not send it to India, they have converted it into a small plaque and they have sent it to us. And the matrix originated, I said, when I traveled back to Birmingham. It was a South Indian restaurant that they gave me a say off, and that is the initial version of the diagnostic matrix. This is Celia Moss and Fiona and Malobi from Birmingham. Yeah, thank you. And then the second component was, while there is clinical variability, the second component was there is genetic variability. So if I have to genetically characterize, there are close to around 16 genes that are currently implicated. Earlier it was 19 genes. So 18 genes, new gene KLSL24 was identified, it is 19. And now CD151 is also implicated. That makes it 20. But after the consensus classification, it has come down to 16. They're close to around 568 exons that you need to screen if you have to identify a particular mutation for EB. Well, this is extremely challenging if you have to do with the conventional route. Genome diagnostics, especially in genodermatoses or across any genetic skin disease, have evolved from the initial Sanger sequencing to now what we call the third generation sequencing, which is nanocode. Right in the middle is where the next generation sequencing has come with targeted panels, whole exome sequencing, and whole genome sequencing. Basically, targeted panels is you will be looking only at a set of genes. Whole exome sequencing is the entire protein coding component, and whole genome sequencing is both the exons as well as the introns. While there are advantages and disadvantages to each approach, whole exome sequencing is currently in the clinical space due to its reducing costs, ability to identify novel genes and dissect out novel phenotypes. There are well-established pipelines for exome sequencing, and the data that you generate is currently amenable to clinical decisions that you can take. While genome sequencing is also in the same route, it is still coming now. There is still some time for the genome sequencing to come into the clinical space for regular diagnostics. Currently, I still feel it is in the research domain. While the third generation sequencing is fastly evolving, currently there is a high IR rate, or I imagine a future where the third generation sequencing will take over the next generation sequencing. With respect to exome sequencing in genodermatoses, it started somewhere in 2009 in the research domain, and in 2020 until 2023, there are almost like 166 new genes are identified with multiple gene cases in almost 63 cases. We could contribute to two. One is the KLS24, the other is Filagrin2, both from Netherlands. And among the approaches, epithelial adhesion disorders enjoy the highest diagnostic rate. This is because of a lot of researchers across the globe contributing to the data, so that we can today, epithelial adhesion disorders or characterizing disorders, we can accurately identify genetic causes in them. So if you take an overall spectrum of genodermatoses, it falls down to around 48%, primarily because lack of multiple studies characterizing them. But most studies have documented the use of whole exome sequencing. While this was there, at that point in time in 2013, as Professor V.R. Rao has introduced already, Sridhar Sisubbu and Vinod Skaria are an amazing team at IGIV who have come up with this program called Guardian. So Guardian was basically to use genomics to understand rare diseases, and they were coming to AIIMS and telling about the program at that point in time, and immediately walk up to them saying that, sir, I have Epidermolysis Bullosa, I have developed a cohort, is it possible for you to teach me how to identify diagnosis? And they have graciously trained me in understanding how to apply exome sequencing and all of it. Currently, Guardian enjoys close to around 280 collaborators across 70 major centers, and it has currently impacted over 5,000 families. Unfortunately, they have recently moved out from IGIV, and they are in the process of setting up their own organization. While with the help of Guardian, we have currently at that point in time, we could characterize the first EB cohort with phenotype-genotype correlations. This is the distribution of the patients that I had at that point in time. While one would think that normally we go to abroad to learn, in my case, the opposite happened when these are the India-specific NGS studies that have been conducted in EB, where initially we have contributed, and recently two more groups. One is Rahul Majan sir from PGH Chandigarh. He's doing an amazing job in Chandigarh. And Ravi Hirumangalar group, now looking after by Divya Gupta from back in Center for Human Genetics in Bangalore. They're also doing an amazing job with Epidermolysis Bullosa right in Bangalore. So you have a center in North and you have center in South that are currently taking care. Both of them recently came up with their publications on their spectrum of Epidermolysis Bullosa. While for me, when I went to Netherlands, because of what I could gain, we could change this traditional approach to a next generation sequencing approach, where Marcel, who is my mentor, has developed this expertise center for EB in the Europe, where he has over 30 years, he has characterized close to around 300 families. So we could actually, for the first time, predict what is the actual prevalence of Epidermolysis bullosa if you consider genetic diagnosis. Earlier, Yo David Fryne from US has given the prevalence rates of 1 in 50,000 individuals, but we think that the prevalence rate is now more, and it is no longer 1 in 50,000, and it is more common than previously assumed. At the same time, we could identify the KLSL24 gene that was identified from German and Chinese groups. We have characterized that it has an important syndromic association, and it is not only a cutaneous phenotype. So patients that you identified with KLSL24, you need to preemptively screen them for cardiac defects, because majority of them, they develop cardiomyopathies, and they are more prone to develop sudden cardiac deaths. So they also have some neurological manifestations, which are currently investigating. And taking the study after his death from to Stanford, we had the fortune to now characterize through another patient cohort that is developing in the US, right from the west to the east, where we see that in dystrophic EB specifically, if you see the pseudosyndactyly, has three manifestations. Either it is not there, partially, or complete. And with the help of Peter Marinkovich, we could identify an antibody which specifically targets the NC2 domain, and this mediates the phenotypic severity. That if you have a staining, you can actually predict what is the severity of the dystrophic EB patients. Conventionally, the antibody that we use is not very predictive, and hence the mismatch in the phenotype-genotype correlations that we have. So, well, this is very soon coming out online. While that has been happening, the other component from that is all the diagnostic side. From the research side, junction EB, as I said before, most of the patients die by 6 months of age. The mean survival is calculated to be around 5.8 months. There is a high prevalence of sepsis. We really don't know why they die. At this point in time, I would want to take you back to my initial experience in UK in 2014 when I was taken to a hospice care. I'm not sure if everyone knows about what a hospice care is, but at that point in time, I was not aware of what a hospice care was. So it was basically a center for terminally ill patients that you know that they are going to die, so that they can go into the most beautiful surroundings and spend their last days. So after finishing a patient visit for a junction EB, I was taken over here, where we were trying to book a room for the patient to come and die. So my return back during that time, again, how a clinician would think at that point in time is what I'm trying to highlight. Is my question was, while my initial thing was, I was not happy with just saying that you are going to die. In UK, which is much more established, even the best that they could do was book a room for a patient to die. I was not okay with that thought again. So going back, instead of getting overwhelmed with it, my question was, why is it so that a patient who is born with this almost the same disease and the same fragility like simplex or dystrophic EB should die by 4 to 6 months of age? What happens at that point in time? My clinical training has immediately posed a question to me, is that the innate to acquired transition for immunity happens at 4 to 6 months. Is there a timeless defect in them? So when I looked back at literature, I couldn't find much. So I asked a professor who was in Birmingham, who spent close to 25 years understanding the thymus development, and he has shared these images where beautifully the laminin 332 also expresses at the basement membrane in the thymus, where it is important for surviving of the developing thymocytes. That means you are developing your T lymphocytes. For them to get educated, laminin 332 plays an important role. Thus came the question when I came back to India. In a junction EB patient that we saw, we immediately did a CT scan and we found hypoplastic thymus. And when we did peripheral flow cytometry, we identified that the immune cells were low. It was a eureka moment for me, but immediately my professor was, this is n equal to 1. It is just a chance possibility. So yes, that is totally true, because patients end up having a lot of sepsis because multiple reasons. And sepsis in turn can also cause a hypoplastic thymus and can also reduce the immune cells as well, because that usually happens in sepsis. So in this hypothesis, when I went to Netherlands, I had the fortune to follow up a patient who we knew prenatally that he is going to be born with a severe junction EB. So as soon as he was born, we looked at a perinatal ultrasound in which this was a thymus at birth. At 1 month of age, this is how it has shrunk down. So at birth, the thymus was normal. But immediately by 1 month, the thymus became hypoplastic for whatever reason. at birth in the cord blood itself, the immune cells are very well present. So for whatever good reason that they are born with a normal thymus, the thymus was undergoing hypoplasia. So when continuing the study, when I went to Stanford, I had the mice model because this was the only existent mice model that was there. Because if you knock out laminin 332, it is embryonically lethal. The mice die by 6 days. So there was a transgenic model that was developed where you can control the expression of laminin by using a simple tetracycline operator drug. So if you give tetracycline, that is doxycycline, they express laminin. If you remove doxycycline, they do not express laminin. So in this, the moment you take off doxycycline, you see that the thymus is getting hypoplastic as compared to the normal thymus over here. So in histology, we observe the same thing. And on expression patterns, we did see that the moment you take off doxycycline, that means laminin is getting low. That means laminin, for whatever good reason, is actually influencing the organization of thymus. And our hypothesis was that laminin 333 mutations not only causes a defective basement membrane in the skin, leading to fragility, but also an abnormal thymus, leading to defective immunity and hence sepsis and hence lethality. For the initial study, I was awarded from IIT, and then for my later studies from EBRPA Patient Organization has graciously given some funding to understand this in a much more holistic manner, and we are currently developing a therapeutic approach for the same. While this is going on the one side, bone marrow transplantation in 2009 has hit everyone with a landmark paper in NEJM from Jacob Tolar from University of Minnesota, where he said that once you do bone marrow transplantation with recessive dystrophic EB patients, there is good survival rates. When I went to Netherlands, I had the fortune to lead this clinical trial where we, instead of doing hematopoietic stem cells, we gave them cord blood stem cells along with mesenchymal stem cell infusions. While there was clinical improvement in the patient, we did not see any expression of collagen 7, nor on immunofluorescence, nor on electron microscopy, and both the patients, they died. Hence, because of increased risks that were there, because of immune rejections and other immune reduction regimens that we use for basically transplantations. So basically, this was the kid that we started, and this was what we made him before he died. And something this was not what we are acceptable with. So we have prematurely closed that clinical trial and published on negative findings. One important aspect that I wanted to highlight from here is, it is very important when you do therapeutic approaches, especially in genetic skin diseases. It is not always important to show the successes. It's also important to show the negativities. Because if you have negative results, it is where you start learning across as well. This experience kind of helped me in helping Peter lead this trial, which is from Crystal, where we used a topical gene therapy. It is a very ingenious approach in which normally for gene therapies, people have used lentiviral vectors and adenoviral vectors. Crystal has come up with a technique in which they have used herpes simplex virus, because herpes simplex virus is highly skin tropic. Because almost majority of patients would already have infections and they would have already developed antibodies, and it is immune evasive as well. So you would normally not develop a rejection against it. So we packaged that entire collagen 730 kb gene into a herpes simplex vector that is non-pathogenic, non-replicative, into a topical formulation and applied it on the wounds. And we see a very good expression pattern, including formation of collagen 7 on immunofluorescence as well as on electron microscopy. It is highly fortunate that FDA has approved this for therapy now. This is the first topical gene therapy that got approved. And also this for EB, at least the advantage is that the whole therapy is in a bottle that can be shipped anywhere. Unfortunately, it costs a bomb currently, and it is not yet available in India, or trials are currently going in Europe, but we still don't know when it is going to be available in India. That gave, when I unfortunately, when my father expired in 2021, is when I transitioned back to India. So my question was, if I had the fortune to do diagnostics, understand some prognostics from therapeutics from the best of the people across the globe, when I come back to India, can I actually give it back to the patient who actually inspired me to take this research? For example, the same old rural patient who has come from a remote village in Uttar Pradesh, can I actually administer these therapies to him? My answer was no. So it still will take a lot of journey for them to actually take it. So hence, my whole concept of this lab that is being developed was, how can I use bedside techniques, bedside observations, take it to the lab, and develop some ingenious therapies and bring it back into the clinics as soon as possible. So I use a multi-omics approach with a lot of biopsies and patient animal models like zebrafish and mouse. And we try to develop a personalized therapy, which is based on revertant mosaicism. I'll come to it a bit later. And some more small molecule therapies. So basically, the idea is use the genetic experience that I have in the Guardian program, take patients that are available across India with collaborations from multiple people, and then we identify the genetic diagnosis in them so that we can immediately enable some genetic counseling or prenatal testing for them. We have currently developed cohorts for AV ichthyosis and some rare genodermatoses, including follicular dowling disease, woolly hair syndromes, autoinflammatory diseases. And importantly, coming back to the topic of today, which is how common is epidermolysis bullosa. Building on the earlier cohort that I developed during my PhD time, we currently see a lot of spectrum across all these centers. Patients have come from all these centers, in which we see collagen 7 to be very common. Is this actually true, or is this just chance observation that you see the severe phenotype that is coming into the patients? Because globally, you see EB simplex to be very common. Is dystrophic EB equally highly common? So we mapped out using population data this time, because IGIB has also contributed to developing the population genomics for the country. So we used indigenous the data that we generated, and globally there is Nomad that is available. And we see that collagen 7 is highly prevalent across all the countries across the globe. But if you look at disease prevalence that has been reported, EB simplex actually occupies a major chunk across all the countries that have characterized it genetically. And dystrophic EB is more or less similar to simplex. Junction EB is in the middle, and Kindler, as Sarachandra sir has already pointed in the beginning, it is quite rare. We mapped out the carrier frequency of these particular variants, and he asked the question is, while we know, are we able to identify these patients? Are there actually more? Because earlier when we said EB is more common than what we previously assumed, we looked at what is the population telling us, and yes, collagen 7 is highly prevalent in the population with a lot of carrier frequency, especially in India. 1 in 114 individuals carry a pathogenic variant for collagen 7. It is not so surprising that we are identifying more and more collagen 7 patients. And yes, in the future, when we do a much more robust screening methodologies, we assume to find a lot of collagen 7 patients. And hence, it is appropriate because a lot of therapies are also being developed against collagen 7, and they can be actually used for these patients. How do I translate this finding to an Indian scenario is the next question that I asked myself. So we came up with a strategy that was evident during COVID. So during COVID, for genomics, they have to identify the variants for COVID, they have developed Illumina has developed a kit called COVID-seq. So COVID-seq is a very rapid genome sequencing approach, and most of the country has enabled infrastructure wherein they have given most of the expertise centers to develop their sequencing capabilities. Suddenly you see a lot of centers that are able to do a lot of sequencing. Can we actually target that infrastructure that is developed instead of saying that we'll do it at IJB? Can we make it more accessible to patients? Is when we have removed that COVID-seq assay, removed the COVID amplicons, and put up our EB amplicons inside it, and said that can we actually do it in a screening approach now, and converted it into a pocket gene. So you have an online version now where you can actually look at your results within 24 to 72 hours. So that helps the clinicians and the patients. So why I call this as an integrated diagnostic and therapeutic approaches. The moment you identify the collagen 7 patients, there is an interesting component that was first identified in mast cell as revertant mosaicism, that is naturally occurring normal patches within the skin in the middle of mutant areas. For example, if this is the mutant area, in the middle you find something called a revertant patch that has normalized because of a somatic event, and suddenly you see that there is a correction of the initial germline defect. So the first question that I asked in my lab is this only specific for EB, or is it present across everywhere? A careful literature survey showed that it is not only in skin, but also in metabolic diseases, blood diseases, and muscular diseases like Duchenne's muscular dystrophy. This revertant mosaicism is a very important component wherein germline defects are getting corrected by somatic events. And these events, now you can convert them into therapy, because all you need to do is identify this patch that is normal, and you take a punch biopsy from it and put it on the wound, and it will immediately heal. So you don't need any in vivo gene corrections or anything. This is natural gene therapy that happens by the patient. So suddenly now imagine a rural patient of epidermolysis bullosa showing a revertant patch, and you're able to identify that patch. You can give it back as a therapy to the same patient because it is autologous, patient specific, no immune rejections, complete molecular revision, and it is cost effective and reproducible. The challenge is only to identify the revertant patch, and suddenly a patient itself becomes their own therapy. You don't need pharmaceutical companies, you don't need multiple things to actually administer a form of gene therapy in them. So how can I actually identify revertant narcissism more commonly is my question. And then we developed this tool wherein we identified this, that there are three possible mechanisms in which revertant narcissism is happening. One is a site-specific manner in which either the mutation, that is A to G, became G to A, or C to T became T to C, or there was a second site mutation that was enabling a better, a short but functional transcript, either through exon skipping or slip mispairing. And then there is another mitotic recombination events that are happening. So to capture all of this, regular exome sequencing, targeted sequencing will not help. It is only the whole genome sequencing that captures the intronic and exonic boundaries and all these areas that will be actually able to identify them. Currently, we are sequencing them at 30x coverage. 30x is not good to capture somatic variants, as already known by a lot of cancer studies that people are doing. Calling somatic variants, you need a very high coverage. So what we developed is something called a long range PCR, where we could sequence them at close to 1500x coverage at a cost effective rate with a rapid turnaround. And we have kind of developed this card. It is called a genome card, where suddenly the patient can carry all the genetic information just like an Aadhaar card for them. This is accessible to both the clinician as well as the patient. It is called what we call as a pocket gene. So this for collagen 7, we have developed this, and previously unsolved cases also, because of the lack of coverage between the exon intron boundaries, we could identify large deletions like 7 base pair deletions from these patches. These are my patients in which we could identify those revertant patches. The whole idea is that we think that in Epidermolysis bullosa, there is a triangular situation that was happening. That is, in a mutant cell with a control division, you have those mutant cells developing. In some cases, because when it becomes uncontrolled, they invariably develop cancer. But in the middle of all of this, suddenly there is a somatic event that is correcting them to leading to a reversion. So we are trying to understand this trijunction as to what is going to drive this particular fate, where it actually continues to develop as a mutant cell or goes into a cancerous cell or comes into a revertant patch. Because once we identify this, we can normalize it much more commonly, and we can actually convert into iPSCs or grafting and can give it to patients as their own therapies. We don't need any viral vectors to actually do any gene therapies for them. So we are currently doing a single cell approach. This is an ongoing study in my lab. While this is what I think is one of the major focus that I think for epidermolysis bullosa in the current context is what is going to be useful. While this is what we are doing with EB, the last couple of slides I wanted to show about other genodermatoses, which is primarily for ichthyosis. So ichthyosis was the other disease that I was very intrigued about, and my mentor initially worked on them. So coming back, we are characterizing our own cohort with whatever is the spectrum. With the patient-wide reports, worldwide reports and our cohort, we find that TGM1 is very common. Leveraging population data, we still see that TGM1 is a very highly implicated gene. It is more common than what we assume. And TGM1 is important because it causes some varied phenotypes, ranging from a severe phenotype, as you see in laminar ichthyosis, congenital ichthyosperm ectoderma, to much more milder phenotype, as in self-healing colloidal baby or bathing suit ichthyosis. So we are very fortunate that Sharat Chandra sir has referred to us to a bathing suit ichthyosis patient, where we have characterized the TGM1 mutation, and we think that this understanding how this bathing suit ichthyosis is happening with the same TGM1 mutation could lead to a form of therapy, because capturing more TGM1 patients becomes important, and hence we use the same approach that we use for collagen 7. Developed a long-range PCR, validated it in a de novo cohort, which was not sequenced before, with 100% sensitivity and specificity. And this, we think, can go into the market, and it is cheaper and readily accessible to every practicing clinician. Why this becomes important is because where we observed during our initial study with Professor Sethuraman, where he identified initially that there was high vitamin D deficiency in these patients. When we treated them with high dose vitamin D, there was a serendipitous discovery that the skin normalized in them. So basically, we gave 60,000 international units in these patients, and the skin normalized, including the ectropium. Why did it normalize? We never knew. So we conducted a study and with taking biopsies for transcriptomes, and we think that there is a pathway that is amenable that we can simply give as a therapy. And vitamin D is as cheaper as 26 rupees that can act as a disease-modifying therapy for them. There's an alternate hypothesis that is going around, which is a TH17 pathway, where secukinumab is very useful in them. This is from Amy Palla's group from Northwestern Chicago. But in a clinical trial, they did see that the response is not so good as anticipated. And dupilumab, which is on the opposite end of the spectrum, is actually also helping in these patients. So we come down to T helper cell differentiation kinetics, which is not very straightforward, because the same cell fate can be driven toward the TH1, TH17, or a TH2 pathway. This is the same pathway that is important for atopic dermatitis, psoriasis, ichthyosis, and we are currently focusing on a very specific molecule that we think can be immediately taken into the therapy for clinical translation. It is currently available commercially, and it is off the market that you can directly administer. So we are doing some integrative analysis pipeline to develop these prognostic markers and therapeutic targets. Basic idea is to take from prevalent diseases towards rare diseases, understand what is happening in them, and can we bring better therapies to the market that are immediately available and accessible and affordable to these patients. So why filaggrin more important? Because as compared to collagen 7 and TGM1, filaggrin, many patients carry these pathogenic variants, and a lot of these patients are found for atopic dermatitis. The reason for showing all of this is if people who are interested would want to collaborate, we are open to collaboration and would happy to work along with everyone. In summary, this is what we do. I would like to acknowledge all my mentors because if they have not trained me in the way that they would have trained, I would not be where I am. Because I think if I've seen it further, as Newton says, it's because on their shoulders. And as always, I dedicate my presentations to my patients because it all started, whatever I did, with that particular question. Yes, can I make a difference for them? So this is how you can reach me. Thank you very much for the opportunity.
Thank you. Excellent presentation, Vamsi. Congratulations. There may be some questions from our delegates. Dr. Sharath Chandra.
Shall I stop sharing my screen?
Yeah, you
please.
can stop. You can stop.
Hello, good evening. Yes. It's a fantastic presentation, really. Is the scene for all the three, atopic dermatitis and psoriasis and ichthyosis, same or different?
No, ma'am. For ichthyosis vulgaris, which is filaggrin, the atopic dermatitis is coming out with filaggrin mutations. Recent mutations are coming up with CARD14. So CARD14 is with psoriasis and CARD11 is with atopic dermatitis. We are currently looking at the spectrum of what are the mutations that are capturing them, because more and more cases with psoriasis or atopic dermatitis, we do see a familial inheritance in them. So we assume that these mutations are playing a role. And also, using population genetics, we identified a very large deletion that is evolutionarily conserved. We are currently looking into them.
And you are developing biologics for these things?
So the idea is not to develop biologics, but the idea is to repurpose these biologics. So where it is developed for already, it is commercially available. Can we develop evidence that will actually be useful for these currently considered incurable diseases? For example, so secukinumab is currently being used in psoriasis. Will all patients benefit from secukinumab? Will ichthyosis patients who have the same TH17 benefit from secukinumab? Not everyone. So the opposite end of dupilumab that is available by Novartis, that is also equally available. So that can also be repurposed in a certain set of patients which have a different mutation. So basically what we are trying to understand is which patient is actually going to benefit from a secukinumab therapy versus a dupilumab therapy based on its genetic problem that is already there. So basically we are not developing biologics, but repurposing biologics. So repurposing commercially available drugs to immediate therapies that are disease modifying.
Dr. Vamsi.
Are you developing any ointments, external application?
So one study that we are going is for a topical enzyme replacement therapy for ichthyosis. So that we are developing in collaboration with a CSR institute, which is CMAP, which is Center for Medicinal and Aromatic Plants. They have recently come up with a psoriasis screen, and we are trying to help them to understand who is going to benefit from them. Yeah. So that is currently under research.
Dr. Vamshi, one of the participant wants to ask you, what are the samples that we have to send from an EB patient for proper diagnosis?
So immediately I would suggest that for any if for genetic diagnosis, it would be important to send a trio sample. A trio meaning a patient sample who is affected and their immediate father, mother, if they are there, or affected siblings. Because when you do exome sequencing, the moment you identify variants, you need to do a familial segregation studies. So having a trio would benefit. So the mandatory would be patient sample with parents, and if available, affected siblings, 3 ml blood in an EDTA vial.
Yeah.
Dr. Vamsi.
Yeah.
Yes,
Yes,
yes, doctor.
Yeah,
Sorry.
when you just spoke about repurposing the biologicals or other drugs, I recently happened to have a Netherton syndrome patient, which you had done the mutation analysis. And we have tried, because of the ease of availability and cheaper cost, we have tried tofacitinib for this patient, and happy to share that it is working wonderfully. After 1 month, the patient is showing almost 80% relief from his symptoms. The ictiosis linearis circumflexa is almost nearly gone, and we are following him up for further course.
that is excellent, sir. That is excellent news. I think what we consider to be incurable before can be actually treated properly with a proper available drugs. Is what I feel.
Yeah.
Excellent.
And, yeah,
Yes, sir.
1 second, sir.
Yeah.
And you had mentioned that this Kelch-like 24 molecule thing, we have to look out for cardiomyopathy.
Correct.
Yeah, so for any EB simplex patient with more than localized forms, with a little bit of intermediate severity or something like that, do we need to go for this screening of troponin T or fibroblast binding protein or 2D,
Sir, it would
3D?
definitely help, because more than troponin T, atrial BNP, that is brain natriuretic peptide, will definitely help in enabling them. But not only simplex patients, very important is woolly hair syndromes. Woolly hair syndromes, you have a lot of like desmoplakin, placoglobin, and placophilins that are happening. They are also prone to invariably develop cardiomyopathy, because there is 100% penetrance. So in all of them, you can actually preemptively screen for preventing sudden cardiac death, where actually dermatologists can be lifesavers.
Yeah, okay.
Sir, Professor Rao?
Any more questions?
Dr. Vamsi, excellent. Revertant mosaicism. No?
Yeah.
Is it necessary to go for the mutation-specific type of therapeutic intervention? When I identify the patch, okay, with revertant, if I am able to identify, for identification of that, I have to go for whole genome sequencing, single cell-based sequencing.
So for identification, we are thinking that our amplicon sequencing will definitely help, the technique that we developed, right? So where we are trying to identify the mutation, I think that taking a biopsy and taking blood, so blood would act as a germline, and in the skin would act as a somatic variant. We could reliably identify the somatic variant, and immediately we can take it to a punch biopsy and a therapy. What we are using for single cell is to understand the mechanism of how the reversion is happening,
Yes.
so that we can develop it.
I need not wait for the total whatever results you are sequencing and all that. That is the research part. But as a punch biopsy, I can try. That is possible. People are doing that.
Immediately.
And
Immediately, the moment we identify a somatic variant,
yeah.
it is possible to immediately take it back to the patient.
But how to identify this patch? You didn't say that, by visually or.
So currently, we do not have any straightforward guidelines to identifying it. Experience says, and also literature says, that within diseased areas, earlier it was a diseased area where there is a lot of scarring and everything. Suddenly, you will find a normal looking patch. It could be with a mottled pigmentation or a apparently normal looking area. So if I can just share my screen once more. You're able to see my screen?
Not yet.
Not yet.
Yeah, right
so,
now it's okay.
Yeah. So
now
you would
we
look.
can see.
So this is an area that is all affected. Within that area, this is also developed vitiligo. Within that area, you would see this apparently normal looking area. So even over here, if you see over here, you would observe that. you would observe these normal looking patches within the diseased area. So this is
Okay.
what we are currently looking at as a possible revertant patches.
Okay.
So in the clinic, we can observe ourselves and also ask the parents to look for
Yeah.
such patches keenly,
Correct.
right?
Correct.
So
Yes.
recently I had one junctional EB patient.
Mhm.
So after speaking to you, I specifically took all the photographs of diseased areas, and I asked the patients also to look for any revertant patches where the skin is not getting involved again and again.
Correct.
So I think
So
right now we can do that and then see for the results.
correct. So in junction EB, more common is collagen 17. Invariably, Marshall's group has already shown that collagen 17 patients, invariably, all of them develop revertant patches. So now this
Yeah,
is more
then,
common with collagen 7s, laminins, keratins as well. So it is an evolving mechanism where with more and more awareness, we will be able to identify more and more patches.
yeah.
Excellent.
Thank you. So any more
This,
questions from the audience, please?
yeah, Dr. Vamsi,
Yes, sir.
you have raised a very
You can call
important.
me once you want this line.
I say what is happening. What is needed is performing science. We call it performing science, and the patients, whoever referred to Genome Foundation, we do all genetic tests and all that. The clinic we share with clinicians, and we did find 99% diagnostics we are able to see. But the
Correct, sir.
question here is the affordability of the test. Now you say you have a research fund. That's what I am understanding, and
Correct.
you will be doing whole exome, whole genome sequencing as a research part. But my earlier experience for whole genome sequencing with a research organization was like this. They said they can't share the results unless the research is complete. They are responsible to their funding agency, not to the patient, not to whoever I have advocated for it. So this is a very critical mass. Research is one place, but what we expect, there has to be performing science. You should be able to convince your funders that whatever you're finding, today you put some test, even, whatever results you have, immediate translation to the patient. Unless until that is not done, whatever we are talking and research, excellent, ultimately the products which we are talking, it is going to be a public-private partnership. Okay, even IGIB, I know they have lined up, a lot of companies they're collaborating. Okay, now tell me this type of, gene therapies which have come, whatever they have taken, the samples for viral vectors, it is very costly. They developed it, but ultimately they are selling, to a huge cost to our, especially third world countries. Mind you, our country has got in terms of genomic ancestry, our ancestry of these mutations goes back to 40 to 50,000 years.
Correct.
Understand? And you compared to all over the world, we will have a cohort. If you were able to sample from different parts of the country, you will have best cohort with lot of variability, lot of genes. For example, I tell you, yesterday only we received reports of one patient. He was identified, diagnosed as EB simplex. But what we found was peptidase D deficiency. Okay. And when I looked at the genes, whatever we have mentioned, it is not there. This peptidase deficiency gene, PEPD gene. Okay. So a lot of genes are going to be discovered. Lot of recalibration is going to happen unless until we have a strong epidemiology, which is, India, whatever patients and clinicians, you should, at this time, with your energy and with your, enthusiasm, you should develop a center, where we are doing the therapeutic interventions. We are discovering. We are innovating methods for genetic engineering. Genetic engineering is very innovative type of things, what is happening, the vector or non-vector, you mean things in terms of epigenetic modulations, for these genes. So a lot of even CRISPR-Cas9, it is an innovative, simple method, but unfortunately, these innovative methods are going into different direction of commercial, exploitation. So I would like to see, like you have in eye disease ophthalmology, okay, you have excellent center with all ophthalmologists, LVPI, they have excellent genomic lab within themselves, okay, and earliest clinical intervention is in hematology.
Yeah.
I was in Bombay. The hematology, they have genome lab. All the hematologists, okay, they have their own center for genetics, like that, because our country is very wide and all that. We have islands of excellence. I call it islands of excellence, CCMB, IGIB, and Institute of Science. Whenever I interacted with them, I have collaborated with Subbu, and but the question was the science funding for them. Their objective is different. My patient, ultimately has not been helped by those tests. That is what very critical mass you have to address, and you have to.
I totally
See,
agree with everything, sir. For me personally, I don't see. So there is a very clear question that I try to address. Is that for me, at least for me, clinicians are not sample providers and researchers
yes,
are not result generators.
yes,
So it has to be a continuum between the both of them, that they have to work together in consensus to identify what is best both for science and for the
yes,
patient, where patient becomes the priority. So for me, my approach is very simple, that whatever patients that I get currently, yes, it is funded by our research organization. While that does, my address to my research organization is very simple. My tangible return for them is to able to identify a patient with a definitive diagnosis.
yes.
That itself is a tangible return for me. And for
Right.
me is to give back what is as a report. So what we cannot give is a clinical report as a research organization. We give what we call as a research report. And with research report, whatever findings that I have, irrespective of whether it is positive or negative, I already share with the clinicians.
I tell you one example. Patient came from USA, okay, and he was looking for prenatal diagnosis, and the boy is already suffering.
Yeah.
And IGFA took up the sample and they did whole genome sequencing and all that. This man was expecting, sir, give me a report so that I can go for my wife's PND. Okay. They said, we cannot give clinical report. We will give you whole exome report. You
So
take it.
at that point in time, I understand the challenges is that
This man, without knowing, the critical mass is here is that he wants a clinical report so that he can show in USA or he can come back to India to get your pregnancy, tested. These things, in fact,
sir,
all these patients who come to us, first and foremost is diagnosis, and another is prevention. She's carrying now, and she wants to know. If I go to a research lab, I am stuck up. I have got with personal experience I'm sharing with you. Okay. So please look into this aspects. But one thing, once I tell you, you should, with these groups and other groups, Dr. Sharat Chandra, lot of dermatology colleagues, you have to think in terms of organizational, some efforts in terms of let us say you are having a database, not only the data, okay, but samples, okay, you have to store the samples, okay, let us say DNA banking. That has to be founded on principles of ethics, okay, everybody should accept, and such type of guidelines in the IGAB you should come out with such type of arrangement, okay,
sir,
where
why only IJV, sir? I think
as you
India
said,
is a unique country with its unique problems, and we only have to bring up our unique solutions. And it can only happen through collaboration and trust. And
yes,
I personally feel that at the end of the day, patient is of utmost importance. And for me, I only see that is the patient actually getting a diagnosis, and am I able to work with the clinician to actually see how better a therapy can be done. So I'm not looking at futuristic goals. While my one foot is in a futuristic goal where I want to develop gene therapies and cures, but my realistic point is how can I immediately help in disease modifying? Can I actually do anything that will actually help them in having a better quality of life? So that is immediate goal is to bring diagnosis. The medium goal is to have a disease modifying therapy, and the long term goal is to have curative therapies for them. So yes, working in that direction.
Your matrix is excellent, EB matrix. Why can't you the database of, let us not go into any other testings and all that. You should take the platform, this platform, dermatologists and all the dermatologists, we can bring out a national platform. Try to understand what is the disease burden, why our policy on rare diseases, this skin disorders is not there, rare skin disorders.
Correct.
That we should address. I am asking our society, the association colleagues to form a committee and try to look into what are the policy guidelines, how we can project, our the disease, skin diseases, rare skin diseases, and the trauma and the psychological trauma, economic, burden for management. Why not government has to include in Arogyasri or some other schemes? We have to create, some awareness among the politicians also, governments also. Okay, and you are in a tech lecture today, fantastic. You have a lot of dermatologists listening to you. Okay, this type, we need to have more and more in future, and also some organizational effort, in terms of forming some body, not that government body. It is something.
Sir, I think it always all starts with local. I think like Sharat Chandra sir has already pointed out. So Sharat Chandra sir, I met him in a conference, and he from there he calls and says, "I have a patient. Do you think you can work on this?" I said, "Yeah, sure, why not, sir." It took us some time, but eventually we could do, and hopefully we'll continue working more. So the whole idea is it all starts locally, and then it can become nationally. But somewhere something has to begin, and it always starts with collaboration and trust, and see what is benefiting for the patients. So
Agree.
Yeah.
Thank you.
Thank you once again.
That's all, sir.
Yeah,
Any
madam,
more observations?
Perfect. Then we call it a day.
Gargi, vote of thanks. A bit of vote of thanks by Gargi.
This was such an amazing session. Everyone was enjoying the presentation, the information. We could hear the excitement in everyone's voice, even in the discussion. It was really nice, sir. And got to learn a lot of things. So glad to attend this session. Thank you so much, Vamshi sir, for your educational talk. And thank you, Rao sir, for chairing the session. And thank you, Sharat sir, for convening the entire event. We are hopeful that the information passed on is going to be helpful to everyone, and more it will spread to the others also, and more such helpful talks, helpful information will be conveyed to everyone. Hopefully we'll all be able to collaborate even further on other topics also, and make sure that this also becomes even more successful. I hope no one has any more queries. If anything is there, please drop it into the groups or in the chats, and we can accordingly follow up with that. If anyone has any concerns, please do let us know. Any feedback, positive, negative, all criticism is accepted. Thank you so much for your attention.
Thank you.
Thank you very much. Have a good rest of the day.
Thank you, everybody. Thank you.
Thank you, everyone. Have a good day. Have a good day, everyone. Thank you. Good night.
Thank you.